To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So I'm going to continue with inheritance. In the previous chapter, we looked at gene disorders, genetical disorders that are inherited from generation to generation. In the previous chapter, I discussed two disorders. One is hemophilia and the other one is red-green color blindness. Both these disorders are caused by genes linked to the X chromosome. So, sex-linked disorders, sex-linked genes that cause the disorders. In both the disorders, the genes that were responsible for the disease condition were linked to the X chromosome. So, then I told you all there is another type of disorder which occurs due to gene mutations. And that is what we are going to discuss now. So, gene mutations and related inherited disorders. So, when we say mutation, what is that? Now, you have the normal genes. Genes are part of DNA molecules. Now, you all know a DNA molecule is a polynucleotide chain. Two strands of polynucleotide chains combine together and they form the double helical structure which was discovered by Watson and Crick. So, that is a chromosome. So, chromosomes are made up of DNA strands. In a DNA strand, a polynucleotide chain that is made up of nucleotides. When we say nucleotides, you all know a phosphate group, nitrogenous base and a pentose sugar. The pentose sugar is deoxyribose sugar. And there are four types of nitrogenous bases: adenine, guanine, cytosine and thymine. Out of those, usually adenine pairs with thymine, cytosine pairs with guanine. We have discussed all those. So, there is a certain sequence for this nitrogen base pair that occurs in the chromosome, and that particular sequence determines a particular character, and that is what we call as a gene. Now, a gene that is responsible for a certain character. Let's say the production of melanin, the pigment that is present in our skin. Now, because of these pigments, the skin has a certain color. If the pigment is absent, the skin will become white. Not the fair skin, but it will almost become like white. No pigmentation at all. Even the hair, the eyelashes, eyebrows, everything will be white. So, that is due to the absence of this melanin pigment. Now, why does this melanin become absent? The chromosome that has the gene responsible for the production of melanin, the pigments, gets mutated. There is a change. The base pair sequence is modified. So, because of that change, the particular gene does not make the pigments to be produced. So, that is what we call as mutation. A change that occurs in the gene responsible for a particular character. Now, don't confuse this with a variation. Now, you all can remember, students, when meiosis occurs, during the production of gametes, meiosis occurs. You all know during meiosis, the number of chromosomes become half, and also there is a possibility of variations. Now, there, what we say as variations again changes to the combination of genes, but that leads to the production of new characteristics. So, that is good for evolution. A similar thing, but here there is a change in the gene or maybe the nitrogen base pair that leads to the modified gene. And that particular mutation, that particular modification is what we call as mutation, and this mutation can lead to disorders. So, when this mutation takes place, that is, a change occurs in the genes, that can lead to a disorder. Then, of course, it becomes an undesired effect. Not that it's undesired, it is there in the population, but the person with the mutated gene will not be 100% healthy. So we will be looking at a few of those such conditions. Or the disorders, inherited disorders due to gene mutations. Due to a change in DNA of a chromosome, the mutations occur in a single gene are known as gene mutation. So that is what I explained to you all. So this is due to a change in DNA of a chromosome, the mutations occur in a single gene are known as gene mutations. So when a naturally active gene is mutated, it gets inherited. We will look at few genetic disorders that occur due to a mutated gene. So a naturally active gene, like I said, the gene responsible for the production of pigments in the skin. So that is an active gene. If that gets mutated, then it gets inherited to the next generation. So these disorders that are inherited occur due to gene mutation. So how does the gene mutation occur? Now we will look at that. This is a do you know box. So occurrence of gene mutations take place due to several reasons. Now the gene mutation can occur due to different reasons, several reasons. Sometimes they occur spontaneously due to no reason. When there is the production of DNA inside the cell, when DNA is synthesized itself, there can be a change. That can due to mutation. Something that is unknown causes the change in the DNA. So that is what we say as spontaneous without any external effects. Nothing affects it. Internally, while the chromosomes are being produced, while the chromosomes are being synthesized, that is the synthesis of DNA molecule itself, there is a change. So that is a spontaneous mutation. Then there is this radiation. Due to radiations. Now when you visit the hospitals, you would have seen different radiation areas. They use gamma radiation, they use X-rays for X-ray photography. Then there is the UV radiation that comes from the sun. So in hospitals, you might have seen this gamma radiation and the X-rays. In that, you have seen that sign which says there is radiation. And they normally say, do not enter without permission or without purpose, or it's only for the official personnel. And especially, they mention that if a person is expecting a child, a mother carrying a child, then not to undergo these particular X-ray photography or radiation treatment. And also normal healthy people not to enter unnecessarily. Why is that? Because these radiations can enter into our body and they can mutate the genes. Same goes for the UV radiation. Even the UV radiation can cause gene mutations. That is why they can lead to cancer. Skin cancer, you all are familiar with that. If we are exposed to too much of UV radiation, it can cause skin cancer. It can cause cataract. In addition to that, it can cause skin cancer also. So those are radiations. Radiations can lead to mutation in genes. So we can have the gamma radiation, gamma rays or X-rays. These are all radiations that can cause gene mutations. Then there are some chemicals, due to chemicals. Now, we use different chemicals in our day-to-day life, even as cosmetics. For example, the hair dye. Some of these hair dye, not all, some of them can lead to gene mutation. Not only hair dyes. There are so many other chemical substances. I'm not going to go into detail there. For example, there are chemicals called as carcinogens, cancer-causing agents. Chemicals that can induce cancer. can cause cancer in a person. Even those carcinogens cause gene mutation. So hair dyes, hair colors, carcinogens, these are all chemical substances that can cause gene mutation. So this can happen in 3 ways. We have discussed 3 ways. There are several ways, but we have discussed 3 ways. One is the spontaneous one, otherwise due to radiation, or it can be due to chemicals. So all these can change the gene. It can change the DNA molecule of the chromosome. So because of that, the gene will get mutated. Is that clear to you all? So with that, students, I'll move on to the next slide. The first disorder that is caused by mutated gene. So like I told you all, the pigment responsible for the color of skin, that is produced only when the gene is the normal gene. But if the gene is mutated, then of course the pigments are not produced. That leads to a disorder called albinism. So here, if you look at the picture first, look at this person, the hair is white, and even the eyebrows, the eyelashes, everything is white in color. The skin, you can see a difference. Like I told you all, fair skin is different from a skin of the person who has albinism. You can see differently. White means we don't normally take the normal white like paint, but you can see a difference. Here, everything is very pale and white. Not only people, even animals can be albino. Now, this is an albino peacock. Now, look at the feather of the peacock. Normally, you all know the peacock feather has many colors. Blue, green, greenish blue, different shades, and it's a nice design. But here you see the feather of the peacock is completely white because that is an albino peacock. There can be albino snakes. So like that, people as well as animals can be albinos. That is because the gene responsible for the production of pigments that give rise to the color of skin is absent or it's mutated. The pigments are absent because the gene is mutated. So here, natural complexion is due to a pigment called melanin. So you have to remember that. It is due to a pigment called melanin. Now this disorder occurred due to a mutation of the gene which is responsible for the production of the above pigment present in autosomal chromosome. Now this is present in autosomal chromosome. You all know what they are. In man, there are 23 pairs of chromosomes. The first 22 pairs, they are known as autosomal chromosomes. And they are responsible for the characteristics of the person. The normal characteristics, physical characteristics of a person. The last pair, that is the 23rd pair, that is the sex chromosome that determines the sex of the person. But already we have seen not only that, the 23rd pair of chromosome also has other genes responsible for other characteristics. But autosomal chromosomes, they contain genes that are responsible for the normal characteristics, the general physical characteristics. So then, the features of this disorder are the abnormal white color of skin, hair, and eyelashes. So it is an abnormal white color, not the fair skin, an abnormal white color of skin, hair, and eyelashes. Albinism occurs when gene is present as recessive homozygous condition. So it has to be a recessive homozygous condition. Both the chromosomes, so it's a pair of chromosome, autosomal chromosome, both have to be recessive. Then the person becomes albino. Not only human but also animals become albino. So another term. you refer to a person or animal having the disease as an albino person or albino peacock. So albinism is the disorder. So this is a genetic disorder carried from generation to generation. So we discuss this under rare inherited characters as well. So it is a character that is inherited due to a mutated gene, and it is a rare inherited character. So it's not common in the population, but you do see people and animals that have albinism. Is that clear to you all? So these are the things you have to remember, students. Now there are disorders due to gene mutation. You need to know what gene mutation is. Then if we look at albinism, you should know what the condition is. That is the absence of pigment called melanin. So that is absent because the gene is mutated. So that is caused by a gene present in the autosomal chromosome and in the recessive homozygous condition. Is that clear to you? So that is one disorder that you need to remember. With that, I am going to move on to the next slide. So here we have the next condition, thalassemia. Thalassemia is a condition where hemoglobin is not produced enough. Now if we read it, this is a condition that occurs due to a mutation in a gene responsible for the production of hemoglobin. Hemoglobin, now you all know what hemoglobin is. We have the blood. In blood, we have the red blood cells, RBC. RBC has hemoglobin. In that, heme is the iron and globin is a protein. And this hemoglobin is responsible for the transportation of oxygen. Oxygen binds to hemoglobin in blood and it is transported by RBC. So if there is not enough hemoglobin, then what will happen? Even though you breathe in, you inhale oxygen, there is not enough hemoglobin to bind with an oxygen and carry it throughout your body. So the circulation of oxygen throughout the body will decrease because you don't have enough hemoglobin. That condition is what we call as thalassemia. So hemoglobin is a protein, what I explained. Hemoglobin is a protein that acts as a carrier for oxygen transportation in blood. Due to reduction of hemoglobin production in thalassemic patients, the main symptom is anemia. You all know what anemia is. Even if there is iron deficiency, there is anemia. Again, not enough hemoglobin, the transportation of oxygen is retarded. So the main symptom is anemia. Here also, it is a homozygous recessive TT condition is disease. So here T and T is not. So far we have been using the letter capital T, simple T with garden pea plants when we discuss the tall and short characteristics. But here the recessive gene simple T. So when a person has thalassemia, that is simple T, homozygous recessive genes, then the person will have thalassemia. Then you can have capital C simple T and capital T. So this of course will be a healthy person and here the person will be a carrier. So if it is homozygous recessive genes, the person will have thalassemia. Heterozygous condition will be a carrier and the homozygous dominant character will be a healthy person. Now this is irrespective of whether the person is a male or a female. All three are possible in both males and females because this is in an autosomal chromosome. So heterozygous TT condition is the carrier. So that is the disease. Then we have the heterozygous condition, that is the carrier. And there are several areas in Sri Lanka with higher number of thalassemic patients. The reason for that may be the marriages between blood relatives. Again, the same thing. Now, reason for that may be due to the marriages between blood relatives. So, that is something you all have to consider, students. You all have to remember. Now, even the sex-linked chromosomes. Now, the mutated genes. All the disorders, the percentage is high. A possible reason is the marriage between blood relatives. So, even thalassemia can occur due to that. If the person is a carrier, or if both male and females are carriers, then there is a high chance that the condition will be seen in the next generation. So, you have to be careful. So, if we try to write this, if you have both carrier mother, carrier father. So, both of them will not have the disease, but they will have the recessive gene in them. So, here, if both are carriers, capital T, simple t, capital T, simple t. So, the genes are going to be like that. That's also capital T, simple t, capital T, simple t. Then when they combine, what will happen? Now here capital T, healthy offspring. Here you have capital T, simple t. Then we have again capital T, simple t, and simple t, simple t. So, healthy person, carrier, and a person with the disorder, that is thalassemia. Now, you can see both are carriers. In the next generation, you only have one healthy person, healthy child, 2 carriers and 1 child with thalassemia. So that is how when there is marriage between blood relatives, if they have the recessive gene among them, the chances of getting the disease increases. Now 3 people, these 2 are carriers. So the gene is accumulating in the population. And here you have a child with thalassemia. So then again, if there is a marriage between maybe a person with thalassemia and a carrier, then also there are chances. So like that, if there is marriage between blood relatives, there is more possibility that the disease will occur. So that is what you need to remember. For here also, for thalassemia, you can have different types of parents. Now here we have seen carrier mother, carrier father. Then we can have a healthy person and a carrier person, or a healthy person and a person with disease. Like that, you can have different combination of parents. So let's say if it is a healthy person, it can be the male or the female, marries a disease, or I'll say thalassemia person with thalassemia, person with thalassemia. So, a healthy person with thalassemia. Here I am not writing whether it's a male or female or father or mother, because it can be anyone. This is the autosomal chromosome. So, healthy person, both are going to be capital T. person with thalassemia, it's going to be recessive condition. So then what happens to the gametes? Now gametes both are capital T. Here it's simple t. Now when the gametes combine, what is going to happen? Now you can see capital T simple t, capital T simple t, capital T simple t, capital T simple t. What happens? Like the normal Mendelian ratio, all are carriers. So then what will happen? Now since again the same thing, if you marry among blood relatives, now this is from one family, you have carriers. Another family also, the person is a carrier. Then those two carriers get married and then they have children. Then there is a very high possibility that there is thalassemia. Like that, when it keeps on happening, the recessive gene becomes more and more in a population. So the people who are carriers and people who have the disease increase in the population. Can you all understand that now? So here first we saw carrier mother and carrier father, both are carriers. So this is how we get the children. Healthy, then two carriers and a person with thalassemia. Here we have a healthy person married to a person with thalassemia. So it can be the mother can be healthy, father can have thalassemia, or the father can be healthy and mother can be thalassaemic patient. But you all have to remember, students, these are theoretically we are looking at what can happen. But if the person has thalassemia in reality, as we discussed, it is a condition where you don't have enough hemoglobin production. So because of that, main problem is anemia. So you can imagine a female having this anemic condition due to thalassemia, they will not be healthy enough. They may not be able to bear children also. But theoretically, we are discussing this. I'm not saying they can't, but it would be very difficult. Already they have the condition where they themselves don't get enough transportation of oxygen because of the thalassemic condition. So, of course, it is going to be a difficult situation. But if we consider, as we discussed, there are certain areas in our country that has very high number of thalassemic patients. So the possible reason is the marriage among blood relatives. So here also you can see everyone becomes a carrier. So that is between healthy person and person with thalassemia. If you have a thalassemia carrier and a thalassemia person, then again the chances of getting thalassemia will be even higher. So like that we can write different combinations. I am sure you all are able to do that because I have shown you all examples. Depending on the parents, you will get different types of children, different combinations. The genotype will be different. Because of that, the phenotype, that is whether they are carriers, they have the disease or they are healthy, will also depend on the genotype of the parents. Is that clear to you all, students? So with that, I am going to move on to the next slide. So this is again for an extra knowledge. This is also related to gene mutation. Now we saw gene mutation where the genes are modified. Now you saw one example where we had albinism. The gene responsible for the production of melanin pigment is mutated. So because of that, it is an unusual white color of the skin, the hair, eyebrows, everything, eyelashes, all that. Same goes for thalassemia. The production of hemoglobin is retarded because of a mutated gene. Similar to that, when there are fetuses, when there is the combination of egg and sperm, there are also mutations can occur. Now, this mutation is due to change in number of chromosomes. In the previous one, I explained to you all, the DNA molecule will be modified. There is the base pair sequence. There can be a modification occurring in that. So, because of that, the DNA molecule is modified. Because of that, the gene is mutated. But here, because of the number of chromosomes, too many, too little. So, for example, you all know from the mother and father, you get the gametes. Gametes are haploid. That is, they have unpaired chromosomes. And when these gametes combine, it becomes paired, diploid. You are familiar with that. So, that is the advantage of meiosis. The number of chromosomes become half. And when the gametes combine, again the paired chromosomes are there. Because of that, the number of chromosomes are maintained from generation to generation. We have discussed this when we discuss cell division. And also even DNA, RNA, with that also you are familiar. But due to some reason, if the number of chromosomes changes, then also that leads to mutation. So here, mutations can occur due to changes in the number of chromosomes. Now, for example, normally, if the chromosomes are X and X, then it is a female. And if the chromosomes are X and Y, it is a male. But there can be instances where there can be an additional X chromosome, or here there can be an additional X chromosome, there can be an additional Y chromosome. So, there the number of chromosomes vary. It will not be 43. It's not going to be 23 pairs. There is going to be an additional chromosome. So, that is a mutation. So, for example, if you have XXX, this is a mutation. Or you can have XXY. Another possibility is the XYY. All these are mutations. Now, there is an additional chromosome. In addition to XX, there is an additional X chromosome. In addition to XY, there is an additional X chromosome. In addition to XY, there is an additional Y chromosome. So, these are additional chromosomes. Let's say this is additional, and that is additional. So, more number of chromosomes. Mutation, that is the changes in the number of chromosomes. There are different instances where these are observed in the human population. So, if you look at those examples, here you can see, due to the presence of a single X chromosome, that is also possible. These are mutations, or else even a single X chromosome. In XX, one is missing. So, only one, less than the required amount. From here also, if Y is not there, only X is there, that is also a mutation. So, this is another mutation. There can be more chromosomes or there can be less chromosomes. Both are possible. So, here, due to the presence of a single X chromosome, a mutated condition called Turner syndrome occurs. They are females lacking sexual maturity and mental retardation. So, if it is only the X chromosome, they are females. They do have the female characteristics, but they don't show sexual maturity. That means they cannot reproduce. They cannot give birth to offspring. they lack sexual maturity and mental retardation. So mentally also their mental development, whatever the development they have to show for their age, does not occur. So there is mental retardation. That condition is known as Turner's syndrome. A syndrome where a female has only one X chromosome, not a pair of X chromosomes. That is the Turner's syndrome. Then there is another type where you have XXY as sex chromosomes. This mutated condition is called Klinefelter's syndrome. Due to presence of XXY as sex chromosomes, this mutated condition called Klinefelter syndrome occurs. Though they are males, now earlier it was females, here they are males, they are feminized, sterile individuals. Though they are males, they are feminized, sterile individuals. So people with Klinefelter syndrome, they are males, they will have the testes, but in addition to that, they may have a developed mammary gland, swollen mammary gland, like that. They can have feminized characteristics and they will be sterile. So they also cannot give rise to offspring. They cannot give rise to fertile. They are not fertile, so because of that, they cannot give rise to offspring. So that is the Klinefelter's syndrome. So Turner syndrome is seen in females where they lack sexual maturity and mental retardation. They show mental retardation. But Klinefelter syndrome, where there is an additional X chromosome in males. Because of that, they are males with certain female characteristics. That is what is meant by feminized sterile individuals. Then there is the other syndrome that is known as the Down's syndrome. Now, Down syndrome, of course, you would have seen people, it is somewhat common. No, it's not a common character, but we do see in our population, people with Down syndrome. This is a mutated condition due to the presence of 3 copies of 21st autosomal chromosome. Now the first 2, it is the sex chromosome, the 23rd pair. But here it is the 21st chromosome. So these people are usually short and mentally retarded. And even from their features, normally they do have a flat face kind of. The nose is somewhat flat. The people are short and they do have a less mental development. So these are symptoms seen in Down's syndrome. That is also caused by a mutated gene. So this is what you need to remember, students. Now mutation is a process that is occurring normally. Now we saw gene mutation sometimes occurs spontaneously due to no reason. Or else there are other factors like radiation, chemicals, but it cannot be controlled by man. So it occurs. And if there is gene mutation and if there is a condition caused by gene mutation, although they have the disorder, the syndrome, they are normal people. So you always have to keep that in mind. They are like that, they have the syndrome because of this gene mutation. So that is again for your extra knowledge, but I'm sure from this you all will understand the concept of gene mutation. You would have seen people with different syndromes, and I think you will be able to accept them more since you all know that it is a genetic mutation that causes this syndrome. So like how we have inherited the characteristics, they also have inherited the mutated gene that has caused the person to be like that. So with that, students, I'm going to move on to the next slide. So with the previous slide, students, I have completed the disorders that are inherited. We saw the disorders caused by sex-linked genes and then mutated genes. Now I'm moving on to a different topic where we discuss application of knowledge of inheritance. So you have a knowledge of inheritance. We started off the lesson by discussing the different characters, common inherited characters like color of complexion, nature of hair, nature of the earlobes, like that. Then there were some rare inherited characters. Even albinism is one of the rare inherited characters. Syndactyl, polydactyl, those are also rare inherited characters. Then after that, we discussed how Mendel did the experiments and explained inheritance. Then we saw what genes are, chromosomes are, phenotype, genotype, the ratio. So with the modern concepts of genetics, we were able to understand and explain the Mendel's experiment. Then the Morgan's theory, Morgan discovered gene linkage. So we understood that as well. Thereafter, we saw how sex is determined in humans and then the disorders caused by genes, inherited disorders. So here we are going to apply the knowledge of inheritance for our benefit. And that is done in different fields. The application can be given to so many different fields. So application of knowledge of inheritance. Since long time, man has understood that by selecting animals and plants with better qualities can produce quality hybrid plants and animal varieties. So here the main aim is to produce quality hybrid plants and animal varieties. What is the meaning of hybrid? Mixing characteristics, crossing characteristics. You can remember the term monohybrid cross. So they are mixing of characteristics. So here also we mix characteristics to get more quality produce. So that is the reason. So here we have some examples of genetic applications to improve quality of animal or plant products. So here to improve quality of animal or plant products. In that, the first one, cows that can produce higher volume of milk. Now you all know those days each household had a cow that was enough to facilitate or look after the family's needs, enough to satisfy the family's needs. But with the increase in population, with the increase in all these housing schemes and all that, you don't have a place to have a cow, and also the milk produced from cow will not be enough to meet the requirement of the growing population if it was the normal way. So what they did was they used this knowledge of inheritance to have cows or to modify the genes to produce cows that were able to produce higher volume of milk. Then there are hens that produce large number of eggs. Same thing. To meet the requirement of the growing population, you need more eggs. So hens that can produce large number of eggs. Then chicken with higher growth rate. So normally when people buy chicken from the shops, they don't look for a chicken that is very small and not enough fleshy. They prefer a chicken that is larger and that has more flesh. So for that also, they use the knowledge of inheritance. Chicken with higher growth rate. Then crops with high yield or pest resistant. Now high yield, again if it is a rice plant, if the amount of rice that can be obtained from the cultivation is more, it is better. So that is what we say as high yield. And also it can be pest resistant. You know what pests are? Pests are insects that can destroy the crops, agricultural crops. Now, if the plant is resistant to this pest, it is easier. You don't have to use pesticides. You don't have to protect it from pest. So that is more useful. Then fleshy, seedless fruits. We have discussed about parthenocarpy. You can remember. Fruits that are produced without the fertilization process. So we can produce seedless fruits. So here in the diagram, they have showed improved seedless oranges. Like this, we can have seedless grapes, seedless apples. So that is possible because of the knowledge of inheritance that was used to improve quality of animal or plant products. So under this, we will be discussing some of the fields in which this inheritance, the knowledge of inheritance is utilized. So with that, students, I will move on to the next slide. So when principles of genetics were identified, production of hybrid plants and animals becomes a technology. So combining characteristics, producing hybrid plants and animals became a technology. It was first applied among some wheat cultivators in America. So it was applied among wheat cultivators in America. The economy was developed as they have used improved wheat varieties. So there was a development in economy due to the use of these improved wheat varieties. Now in Sri Lanka, crop research centers and breeding centers have achieved a considerable development in gene technology. So research centers, they research and try to find new crops with very favorable characteristics. You are familiar with some of them. The golden rice, drought-resistant plants, pest-resistant plants. So those are all improved varieties. Then large-size fruits or vegetables. So these are the things you need to know. Large-size fruits or vegetables, improved grains and livestock have been used, fulfilling the needs of greatly increasing population. This is what I mentioned to you all. To satisfy the needs of the population, these are needed. If we have not used genetically modified, this is an important term, genetically modified high quality products, we would not be able to fulfill the high demand of food. So we will not be able to provide the food requirement, provide food for the whole population. That is the high demand. Because the population has increased, there is a high demand. We will not be able to meet that high demand if we did not use inheritance genetics to modify genetically modified crops and livestock. Both of them help to satisfy the requirement of a growing population. That is what you need to remember. So here again you can see in the picture improved vegetables and fruits. So they are although they look the same, students. Now this is how even a normal fruit and vegetable may look like, but there are so many crops that have been genetically improved. So although you don't see it from outside, maybe from the size, you can see from the taste. If they don't have a seed, then you can see. But just by looking at it randomly, you cannot say whether it's genetically modified or not. But there are so many fruits and vegetables that have been genetically modified and is very useful to us. So you have to remember that. So with that, I will move on to the next slide. So we discussed genetic engineering. Now you all are familiar with the term engineering. Building, modifying, combining. So we apply that type of process in genetics. So when we say genetics, genetics is based on genes that is present in chromosomes. So this is what happens, students. Now let's say we have a chromosome like that. Now this part of a chromosome has very desired characteristic. Let's say it's rich in nutrients. This particular chromosome leads to a character where the particular plant has a produce that is rich in nutrients. So we want this character to be inserted or combined with another crop, so that that crop also will produce plants that is rich in nutrients. So to do that, what we can do is, we can remove this part, cut off this part of the chromosome that has those desired characteristics. Or let's say this is the gene responsible for the production of insulin. Let's say that. Now you all know insulin is a hormone. Diabetic people need to consume insulin. They need to inject insulin if the diabetes need to be controlled only by the hormone. If the tablets do not work, or with the tablets, in addition to that, they need to get the insulin injection. They need a lot of insulin. So to produce so much of insulin, normally you all know our pancreas produces insulin. So you cannot get the insulin from a person. You can't have donors who donate insulin. So we have to produce it. So to do that, what they do is they cut out the gene responsible for the production of insulin. So here we can have, let's say, the bacteria DNA. So what they can do is they can cut off a part of the bacteria DNA and they can replace it with this particular gene. So here, instead of the gene that was present earlier, now we have the gene that is responsible for the production of insulin. So you had the gene in one chromosome, you take that part and you cut off a part from another chromosome and insert this part. And then you let that to multiply. This particular chromosome, this particular DNA is used to produce insulin. It is allowed to replicate. So many chromosomes are produced and that is used to produce insulin. This is just the basic idea. I have given you all the basic idea how this recombinant DNA technology is carried out. So this is the genetically modified process. You have a chromosome that has a particular gene that is needed for us. You take that gene and you insert it into another chromosome and use that for production. And this new gene or this new DNA, it's not the new one, but it is a DNA that has been modified. The blue color part and the pink color part has been introduced into that. So now this DNA is known as a recombinant DNA. You have removed part of it and you have inserted another part. So recombinant DNA, it has been recombined. So recombinant DNA, and the technology used to do that is known as recombinant DNA technology. I'm sure you all can understand that basic concept. So if I go back to the slide, okay. So genetic engineering. Now here this recombinant DNA technology is part of genetic engineering. New technology is being used to produce a recombinant DNA. So that is the term. I explain what a recombinant DNA molecule, which is formed by combining DNA fragments of different sources. So DNA fragments. This is one DNA fragment. The blue color is another DNA fragment. So both are combined together. This field is known as recombinant DNA technology. This field is highly popularized as genetic engineering or gene technology. Genetic engineering or gene technology. The genotype of an organism can be altered by removing or adding extra DNA fragments into genome. So here this is important. The genotype of an organism can be altered by removing or adding DNA fragments into genome. What is a genome? Now if you take a person, we have 23 pair of chromosomes. In those there are certain genes responsible for different characteristics. So all that together is known as the human genome. The chromosomes, the 23 pairs of chromosomes, and the genes responsible for different characteristics present in those chromosomes together is known as a genome, human genome. So like that, all organisms have a genome, and that can be altered by doing what? Either you can remove, say if there is something that is giving rise to a harmful character. You can just remove it, discard it, and you can use that particular chromosome. Or like we did here, you can add a chromosome. So here of course it's like replacing, otherwise you can add also. You have the chromosome, you split it and you add another part to it. All are possible. So you can remove, you can add, or you can replace it with something else. So all those are possible in gene technology or genetic engineering. And that is the field that carries out recombinant DNA technology. Is that clear to you all? So this is again a basic introduction to what genetic engineering is. And now we will see where this is applied in different fields, or how this is applied in different fields. Food and agricultural field. So example for plants and animals with better qualities that has produced using recombinant DNA technology. So already they have been produced. The first one, weedicide resistant crops. Now what are weeds? Let's say there is a paddy cultivation. So in that area, paddy plant is the plant that we are concerned with, because we are cultivating paddy. But at the same time, there can be other plants that grow in that environment, and because they grow, they will absorb the nutrients that are needed for the paddy plant, or sometimes they can even retard the growth of paddy. So those plants are known as weeds. So cultivators make sure that there are no weeds in the agricultural land. Now to do that, what we can do is we can apply weedicides, chemical substances that will destroy the weeds, that will prevent the weeds from growing. But the weedicides are chemicals. Now if the weedicide affects the crop plant, then that can become a problem. So if we can make a plant or crop that is weedicide resistant, a crop that is not affected by the weedicide, then it's easier. You can use the weedicide to control the weed, but the crop is not affected by the weedicide. The crop is resistant to the weedicide. Then it is more useful. So this is done by inserting a gene obtained from a bacteria. From the bacteria, a bacterium, you get the gene that is resistant to weedicide, and you insert that to the genes of a crop, and then you can produce weedicide resistant crops. The same way, pest resistant crops. I explained this before also. Pests are insects that can harm the plant, the crop. So if the crop itself is resistant to the pest, then we don't have to use additional pesticides. We don't have to spray pesticides. So pest resistant crops. Again, by inserting a gene obtained from a soil bacterium. So these are things you can remember. First one, bacterium, here a soil bacterium. Then rice enriched with vitamin A, golden rice, that you are familiar with. So golden rice. A gene that produces vitamin A in carrot is obtained and inserted into paddy. So here you can see a gene that produces vitamin A in carrot is obtained and inserted into paddy. So you add a gene. You have the gene in carrots, you take that and you add it to paddy. The same way we have discussed when we discussed microorganism in the previous grade. a bacteria, Erwinia urodovora, that is also used in the production of golden rice. So like that, there are different methods. Then we have a tomato resistant to cold weather. Now in cold environment, tomato does not grow very well. But if you can make a variety, which has already been done, that is resistant to cold weather, then you can grow the tomato plants in environment where there is cold weather. So this is done by inserting a gene obtained from a fish living in mud of cold countries. So there you can see. Now earlier they used bacteria to get the gene. Then genes from one plant is inserted into another plant. Now here you can see from a fish living in mud of cold countries. From an animal, genes are taken and inserted into a plant. So for gene technology, recombinant DNA technology, it doesn't matter from where you get the gene from, as long as you have the desired character. Then high productive, that is flesh and milk, cattle, and milk with high nutritious value. So if it is a chicken that has lot of flesh, or cattle with lot of flesh, then cattle that can produce a large volume of milk, large amount of milk, and also with high nutritious value. And also food produced by genetically modified organisms. Those are known as the genetically modified food. So in shops, in supermarkets, you do see genetically modified food. You do have different types of areas. In that you can get these also. You can get the normal rice, you can get the golden rice. Then genetically modified fruits and vegetables as well. Larger fruits, larger vegetables with more nutrients. All that is possible in the field of food and agriculture. I am sure you all can understand that. With that, students, I'll move on to the next slide. So here we look at the field, industrial field. So here also there are some uses. Below are some instances where bacteria produced by gene technology are being used in industrial field. So here it is the bacteria produced by gene technology. The first one, production of enzymes such as amylase. What is amylase? Amylase is an enzyme. Usually it's produced inside our buccal cavity. You have discussed this before. It is present in growing, germinating mung seeds. We grind it, we get the extract, and amylase induces the conversion of, it is an enzyme needed for the conversion of starch to maltose. So that is amylase. Then there is the production of some amino acids. That is to produce MSG. MSG means monosodium glutamate. This is used as a flavor in food, to flavor food. So production of those amino acids that are used to produce MSG is also produced using genetically modified bacteria. Then production of vitamins, cyanobacteria, cyanobacteria. You all know what cyanobacteria is. Bacteria and cyanobacteria both belong to kingdom bacteria. And this is used to produce vitamins like vitamin B12 and E. Then it is used as a remedy to the pollution by fossil fuel combustion and other waste material. Fossil fuel, the different fossil fuels that we use, petrol, diesel, when they undergo combustion. Now they burn in air. So when there is combustion, carbon dioxide and water vapor are evolved to the environment. In addition to that, if there are other substances present in fossil fuels, they are also evolved to the environment. So that can cause pollution. To remove these pollutants, we can use the bacteria, recombinant bacteria. This is something that we call as bioremediation. You have heard this term before, bioremediation. where microorganisms are used in the field or used to remove pollutants from the environment. So that is bioremediation. For that, we can use recombinant bacteria. So as a remedy to the pollution by fossil fuel combustion and other waste material. So to do that, we can use genetically modified bacteria. I'm sure you all can understand that. So with that, students, we'll move on to the next slide. In the field of medicine, medical field, gene technology is used in different sections of medical field. First one, what we have already discussed, production of insulin. A hormone, human gene related to insulin production is inserted into E. coli bacteria, Escherichia coli bacteria. So again, a process similar to what I explained to you all. The human gene inserted into a bacteria and the bacteria will produce insulin. Production of proteins including growth hormones. So proteins including growth hormones by inserting relevant gene into bacterium. Again bacteria. Then use of developed bacteria and fungi in production of antibiotics. So you all are familiar with all these terms. Growth hormones. Growth hormones are needed for the growth of a person, especially the growth occurs in your younger age up to the adolescence age. So within that period, a child grows. So for that, the growth hormone should be there in the body of a child. So to produce hormones, growth hormones or proteins, you can use this genetically modified bacteria. Then antibiotics. Antibiotics are medications that are given to control the infections caused by microorganisms. So that is also produced using bacteria and fungi. Then insertion of genes that is responsible for the formation of arteries in the embryo into patients with artery blockages. Patients to carry out bypass surgery to induce growth of new arteries. So here also they insert a gene so that the artery grows. Now you all know arteries are blood vessels that carry blood throughout the body. From the heart they distribute blood to organs, and there is the depositing of cholesterol, and because of that the lumen of the artery becomes small. Then of course if there is a blood clot, that can lead to cardiac arrest, and also they can have different conditions because the amount of blood flow through the small lumen is difficult. So because of that they might need to undergo surgery. So to these patients, they use these genes to induce the growth of new arteries. Then we have the replacement of the diseased gene by a healthy gene. This method is of course known as gene therapy. Replacing a diseased gene by a healthy gene. Let's say thalassemia, a gene responsible for thalassemia, recessive gene. If you identify that in the fetus level, in the embryo stage itself, you can replace it. Gene therapy. Then use of DNA technology to confirm the identity of a person in forensic medicine. So the last one, in forensic medicine. You know what forensic medicine, forensic science is. If there is an incident, say a murder case or something, or some incident where you need to identify the person, you have specimens, maybe a blood sample, a sample of a semen, a hair, something. So from that you can identify the person. You can match it to another person. For the identification of a criminal, blood sample, semen, hair or DNA of any other part of the body obtained from a site of crime are used. Identification of the criminal by checking the compatibility of the DNA of those samples with the suspect's DNA. So you have suspects, you have the sample, you can carry out the DNA test. The DNA technology is used for that, and then you can match the samples. So that is used in forensic science. So these are some applications in the medical field. So we looked at some applications in the agricultural field, then the industrial field, and now the medical field. So I'm sure you all have a good understanding about how we can use the knowledge of inheritance, the recombinant DNA technology or genetic engineering to develop the producers in different fields, and that becomes very useful to us. So with that, students, I have come to the end of this lesson, and in the next slide I will discuss the summary with you all. Summary. The diversity among organisms in the biosphere is because of the inherited features of each species. Inherited features are the characters that transmit from generation to generation. So in the biosphere on the earth, there are so many different species, organisms. They inherit characteristics from their previous generation, and these genes are responsible for that, and that is why we have so many different types of organisms as well, the diversity. Within the same species too, the organisms possess differences. There are common and rare inherited characters in human populations. The field that study about the way that the inherited characters are transmitted is known as genetics. Gregor Mendel was the first person to carry out experiments regarding inherited characteristics of plants. Mendel concluded that two factors different to each other which are responsible for determining a character of the pea plant. Now all these are what we discussed in the lesson. So this is the summary. These factors that determine characters later identified as genes. One of these is a dominant gene and the other is a recessive gene. So you are familiar with all these terms. Gregor Mendel, then the experiments using the garden pea plant, and then it was identified as genes and the dominant gene, recessive gene, all are familiar to you. The pattern of inheritance of a monohybrid cross can be expressed in a chart. So pattern of inheritance, monohybrid cross. So we'll continue the summary in the next slide as well. The pair of genes responsible for a particular feature can be shown in a gene expression. The externally expressed feature of an organism is phenotype. The gene composition that determines a particular phenotype is called the genotype. The material that transmits features from generation to generation is the DNA in chromosome. DNA in chromosome. A specific nitrogenous base sequence of DNA is known as genes. That is important. A specific nitrogenous base sequence of DNA is known as a gene. And the number of chromosomes in nucleus is constant for a species. The number of chromosomes is a constant for a species. The pair of chromosomes that are similar in length, width and location of centromere is known as the homologous chromosome. The pair of genes responsible for a particular character present in complementary locations of the homologous chromosomes. So here students, we need to continue with the summary. So I'll move on to the next slide. There again we have the summary. The genes that are present in the same chromosome and that cannot be segregated independently are known as linked genes. And you all know Morgan introduced gene linkage. Then the sex of human is determined by the way of association of sex chromosomes during fertilization. So you are familiar with X and Y chromosomes. A zygote with two X chromosomes gives rise to a girl, and a zygote with X and Y chromosomes gives rise to a boy. Genetic disorders occur due to recessive sex-linked genes and mutations in DNA. There is a high chance of getting genetic disorders in children resulted in marriages between blood relatives. So marriages among blood relatives can give rise to children with genetic disorders. The chances are high. There is a possibility of fulfilling the demand for food with genetically modified high quality products. That is what we discussed before. There is a considerable development in agriculture, industrial and medical fields due to gene technology. So you are familiar with all these concepts. I'm sure you all have understood the lesson properly, students. I have discussed all these concepts during the lesson. So I want to discuss the questions with you all. In the next chapter, I'll be discussing the questions from the textbook.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.